Ever have those days where your body just feels, I don't know, adaptable?
Yeah.
Like you can skip a meal without completely crashing or maybe power through a workout even if you didn't exactly carb load.
Yeah, absolutely.
And then other days it just feels like your energy is stuck,
Yeah.
right? Like it just can't keep up with what you're asking it to do.
Mm-hmm.
What if your metabolism actually had this incredible superpower?
Mm-hmm.
This ability to just seamlessly switch between different fuel sources depending on, what you're eating or what you're actually doing.
Well, that superpower is pretty much what researchers are talking about when they use the term metabolic flexibility.
Okay, metabolic flexibility.
Mm-hmm.
So today we're doing a deep dive. We've got a stack of sources here, really anchored by a very comprehensive review in endocrine reviews, and also some really fascinating recent findings highlighted in the journal Metabolism.
Good.
Yeah, so our mission today, let's try and cut through some of the complexity, understand what metabolic flexibility is, at its core. Why is it so vital for health? What actually goes wrong when we lose it? And maybe most importantly, what does the latest research tell us we might be able to do about it? Like, can we improve it?
Right. So basically, at its heart, metabolic flexibility is your body's knack for efficiently adapting its fuel use.
Okay.
Think of it like a hybrid car, maybe. It can run on different fuels, mostly glucose from carbs and fatty acids from fats.
Right. Petrol or electric.
Kind of like that, yeah. Metabolic flexibility is the engine's ability to smoothly switch between those fuels, or maybe even blend them, depending on whether fuel is easy to get like right after a meal, or if you need sustained energy, like when you're fasting or exercising.
Makes sense.
And this idea, that our muscles could adapt their metabolism, especially with exercise, has been around for a while. Scientists like Salton and Golnick were talking about something called metabolic plasticity way back in the 80s.
Okay. Plasticity.
Yeah, but the specific term we use a lot now, metabolic flexibility, that was really cemented by researchers led by George Kelly. They did a key study back in 1999.
Ah, the Kelly study. I've seen that referenced a lot.
Ah.
What was so groundbreaking about it?
It was a real eye-opener. They looked specifically at skeletal muscle because that's where a lot of this fuel switching happens, right?
Hmm.
And they compared people who were lean with people who were obese and also insulin resistant. They watched what happened after an overnight fast and then after they gave them insulin, which signals, glucose is here.
Okay.
And the results were pretty striking. The lean individuals' muscles, incredibly flexible. After fasting, they were burning fatty acids quite happily. But when the insulin signal came, boom, they easily switched gears to burn glucose.
Like they were supposed to.
Exactly. But the obese, insulin-resistant individuals, they really struggled with that switch. Even after fasting, their muscles didn't seem to rely on fatty acids nearly as much as the lean group, and they just didn't transition effectively to burning glucose when insulin showed up.
So stuck, basically.
Pretty much. That impaired ability to switch became the kind of foundational definition of metabolic inflexibility. And later studies built on this, showing, for instance, that lean, flexible people could really ramp up their fat burning if you put them on a high-fat diet, but obese individuals couldn't manage that switch nearly as effectively.
So it really boils down to being able to use the right fuel for the job, whatever the situation demands.
That's it, exactly. And this finding quickly led researchers to zoom in on the mitochondria.
The powerhouses of the cell.
Yep, those guys. They realized that the mitochondria's ability to actually choose which fuel to burn based on what the cell is being fed was just absolutely central to this whole flexibility thing.
But it's bigger than just mitochondria now, isn't it?
Oh, definitely. The concept is really expanded. Metabolic flexibility is now seen as this layered capability. It involves the whole body, individual organs, think liver, muscle, single cells, and even those tiny organelles like the mitochondria.
Some multiple levels.
Right. And it's not just a simple on-off switch for fuels. It involves these incredibly precise, really dynamic adjustments across loads of metabolic pathways. But the core purpose is still the same. It's a fundamental way our bodies adapt to keep energy balanced, maintaining what scientists call energy homeostasis.
Okay.
So matching the fuel supply to the energy demand, whether that's because you're fasting or because of the type of meal you just ate, or whether you're sitting versus running, or even things like changes in the environment, like temperature.
Okay, let's really get into the how then. How does this switching actually happen? How does the body make that decision, moment to moment, which fuel to prioritize? What does that biochemical dance actually look like?
It really is like a complex dance. It's orchestrated mainly by nutrient signals and hormones. We can break it down by looking at two main states, when you've just eaten and when you haven't eaten for a bit.
Fed versus fasted.
Exactly. So first, the fed state. Let's say you just had a meal with carbs, your bloodstream gets rich with glucose, and your pancreas releases insulin. Insulin's the key, right? It
Right.
tells cells, open up, take in this glucose.
Right.
Once glucose is inside the cell, it becomes the preferred fuel. It goes through glycolysis, that's basically breaking glucose down, and then the products from that get processed further in the mitochondria to generate a whole lot of energy, ATP.
Right.
And this is where a classic concept comes in called the Randall cycle or the glucose fatty acid cycle. It's based on lab studies, and it suggests this kind of metabolic seesaw.
A seesaw. How so?
Well, the idea is when your cells are busy burning glucose, they tend to put the brakes on burning fatty acids. And the reverse is also true. When fatty acids are being burned, glucose burning tends to slow down.
So if I've just had that pasta, my body's thinking, okay, loads of glucose here. Let's use this first. We can save the fat for later.
That's a great way to put it, yeah. And the mechanism for suppressing the fat burning is actually pretty clever. High glucose and the resulting insulin surge lead to an increase in a molecule called malonyl-CoA.
A malonyl CoA. Okay.
Think of malonyl-CoA as a traffic signal saying, stop burning fat right now. It does this by inhibiting a key enzyme called CPT1,
CPT1.
carnitine palmitoyltransferase 1. It's the transporter, basically the ferryman, that carries fatty acids into the mitochondria so they can be burned, a process called beta oxidation.
Gotcha.
So if CPT1 is inhibited by malonyl-CoA, fatty acids can't get into the powerhouse, they're blocked. And instead, they get rerouted towards storage, usually as triglycerides.
Stored as fat.
Right. And at the same time, the products from breaking down glucose actually help activate the machinery needed to burn that glucose in the mitochondria. It does this partly by inhibiting another enzyme called PDK pyruvate dehydrogenase kinase. PDK normally slows down glucose burning. So by inhibiting PDK, you speed up glucose oxidation. It's all coordinated to make sure glucose gets used when it's plentiful.
Okay, that's the fed state. What happens when the fuel tank starts running low in the fasted state?
Right, so now shift gears. Maybe you slept all night, or it's been several hours since you ate. Glucose and fat circulating in your blood are lower. The hormonal balance shifts, insulin levels drop, and another hormone, glucagon, rises relatively.
Glucagon takes over.
Glucagon sends signals, mainly to the liver. It tells the liver, release some stored glucose from glycogen to keep blood sugar stable. And it also starts promoting something called ketogenesis.
Making ketones.
Exactly, making ketone bodies. And critically, with insulin levels low, that inhibitory molecule, malonyl-CoA, decreases.
So the brake comes off fat burning.
The brake comes off, CPT1. That gate swings open, and fatty acids can now flood into the mitochondria to be burned for energy via beta oxidation.
Okay, and where do the ketones fit in?
Ketone bodies become really important, especially if the fast goes on longer. Your brain, for instance, is a huge glucose user, needs about 140 grams a day, roughly. Yeah. When glucose gets scarce, like during prolonged fasting, the liver ramps up production of ketone bodies, making them from fatty acids. These ketones, especially one called D3-hydroxybutyrate, can cross into the brain and serve as a really good alternative fuel.
Better than glucose.
Well, the sources point out they're very efficient. Apparently, 100 grams of D-DAX3-hydroxybutyrate can yield slightly more ATP energy than 100 grams of glucose. So yeah, very energy dense.
Interesting.
And ketone production is also tightly regulated by insulin. You need low insulin for the liver to really start churning them out, and also for your fat tissue to release the fatty acids needed to make them in the first place. Researchers are also looking into other roles for ketones now, maybe as fuel during exercise or even as therapies for certain metabolic conditions.
So the body has these really elegant systems then for switching between glucose and fat, depending on whether you're fed or fasted. What about going even further, like serious calorie restrictions? Does that trigger different adaptations?
Oh yeah, that pushes the body's adaptive machinery even more. When you significantly reduce nutrient intake over time, it changes some major signaling pathways.
Like what?
Well, with less insulin and related signals like IGF-1, pathways like ACT get dialed down. This, in turn, activates a group of factors called FOXO transcription factors.
Okay, so.
Yeah, think of them as mobilizing the cell's repair and maintenance crews. They switch on genes involved in things like DNA repair, autophagy, which is the cell's recycling system, kind of like cellular housekeeping and building up stress resistance.
So hunkering down and cleaning up.
Pretty much. Another key pathway is mTOR. mTOR usually senses when nutrients are abundant and tells the cell grow, build stuff. But when nutrients, especially protein and certain amino acids, are scarce, mTOR activity drops.
So less building.
Right. The cell shifts focus from building new things to maintaining what it has and salvaging resources. Again, autophagy gets promoted, and energy-intensive processes like making new proteins get conserved.
Makes sense during scarcity.
Definitely. And then there's AMPK. This enzyme gets activated when the cell's energy charge drops, basically. When the ratio of AMP, low energy signal, to ATP, high energy currency, goes up. AMPK is like a master switch for energy conservation. It flips on pathways that generate energy, like burning fat, and it slams the brakes on pathways that use energy for building things.
The ultimate energy manager.
You could say that. And finally, there's a family of proteins called sirtuins, or SIRTs. Their activity ramps up during calorie restriction. They depend on a molecule called NAD plus SAT, which becomes relatively more abundant compared to NADH during fasting. Increased SIRT activity leads to changes in proteins, deacetylation, which influences gene expression and mitochondrial function. These SIRTs are often linked to the health benefits seen with calorie restriction in studies.
Wow, so CR really triggers a deep metabolic reset.
It seems to, yes. The adaptations are quite profound in research models. Of course, the big challenge is how humans can actually sustain something like CR safely and effectively long term. That's a whole other discussion.
Right. Okay, so that's the system working well, being flexible.
Yeah.
But you mentioned earlier, this system can break down. What does that look like? What leads to metabolic inflexibility?
Yeah, so metabolic inflexibility is when your body just loses that smooth ability to switch fuels effectively. And unlike, say, an inborn error of metabolism you're born with, this is usually something acquired. It often develops over time.
Acquired how?
The primary driver, highlighted again and again in the sources, is chronic caloric excess. Basically consistently taking in more energy than your body actually needs.
Eating too much too often.
Essentially, yeah. When you constantly bombard your cells, particularly your mitochondria, with a surplus of fuel, too many fatty acids, too much glucose, excess amino acids, it can actually overwhelm the system.
Overwhelm the mitochondria.
Exactly. The sources describe it almost like a metabolic traffic jam. They use terms like a push mechanism, where the sheer volume of incoming fuel causes persistent inhibition and confusing signals. It leads to what they call mitochondrial indecision.
Indecision. So the mitochondria get confused about what fuel to burn.
That's the idea. They struggle to make the switch efficiently, and because they're overwhelmed, they don't end up burning either fuel source very effectively.
So what happens to the excess fuel?
Well, it leads to incomplete breakdown, incomplete utilization, and crucially, that excess fuel gets stored, but not always in the right places. It starts accumulating in what researchers call ectopic depots.
Ectopic,
Yeah.
meaning like in the wrong place.
Exactly.
Yeah.
Think fat building up not just nicely tucked away in your adipose tissue, but accumulating within your muscle cells, within your liver cells, places it's not really designed to be stored in large amounts.
It doesn't sound good.
It's not. And it creates problems. For example, remember that PDK enzyme that puts the brakes on glucose burning?
Yeah.
Well, a sustained surplus of fatty acids can actually signal the cell to make more PDK. So having too much fat around can actively blunt your ability to burn glucose effectively. It messes up the balance.
Creates a vicious cycle.
It can, yeah. And inside the mitochondria themselves, this overload causes chaos. If you have lots of fuel coming in, but maybe low demand for energy because you're not very active, you get a bottleneck.
A backup.
A backup. This can increase the production of harmful molecules called reactive oxygen species, ROS. These cause oxidative damage, like cellular rust, and they can mess with important enzymes. Some researchers call this mitochondrial metabolic gridlock.
Gridlock, like traffic.
Yeah, exactly. You get a buildup of certain molecules like NADH. This creates a chemical imbalance. The NADH-NAD plus ratio gets too high, which slows down key energy-producing cycles like the TCA cycle. This then causes upstream molecules like acetyl-CoA and other fatty acyl-CoAs to accumulate right at the convergence points of different pathways, further jamming up the whole metabolic flow.
Wow, it sounds like a complete system breakdown.
It has knock-on effects, for sure. That buildup of acetyl-CoA, for instance, can lead to excessive acetylation of proteins, which can change how they function, not always for the better. And that high NADH-NAD plus ratio, it inactivates those beneficial SIRT enzymes we talked about earlier.
The ones involved in healthy aging.
Yep. those, so you lose their protective effects. And studies in animals fed high-fat diets for long periods actually show a reduction in the expression of genes needed to build and maintain healthy mitochondria.
So the powerhouses themselves start to degrade.
It seems that way.
Right.
And systemically, this reduced ability to burn fat properly leads to the accumulation of specific types of fat intermediates inside cells. Things like diacylglycerol, ceramides. These aren't just inert storage molecules, they act as toxic signals. They directly interfere with insulin signaling.
How?
They can trigger changes to the insulin receptor itself, making it less responsive. They mess with downstream signals like ActPKB. Essentially, they make your cells deaf, or at least hard of hearing, to insulin's message to take up glucose.
Which is insulin resistance.
Exactly. This long-term impairment of metabolic flexibility across many tissues, muscle, liver, fat, is strongly correlated with the development of metabolic syndrome, obesity, type 2 diabetes, and cardiovascular disease. It's a state where your whole internal energy management system becomes dysfunctional. And yeah, building on that, the sources really dig into how this inflexibility manifests in specific diseases.
Okay, like obesity.
Obesity is kind of the classic example, really. Fundamentally, it's a disorder of regulating energy balance and fat storage. People with obesity often have higher levels of free fatty acids circulating in their blood.
Which we know causes problems.
Right. Those excess fatty acids interfere with glucose metabolism, inhibiting key enzymes involved in using and storing glucose, like glycogen synthase and PDH. And often, their skeletal muscle mitochondria just don't function as well. Their capacity for burning fat for beta oxidation seems reduced.
They can't adapt to high fat.
They seem less able to properly activate the PPAR alpha pathway, which is really important for ramping up fat burning when fat intake is high. So when their main fat storage cells, the adipocytes, get full, that excess fat has to go somewhere.
Ectopic PPOs.
Right. It spills over into muscle, into the liver. And this ectopic fat accumulation is strongly linked to all sorts of metabolic issues: insulin resistance, type 2 diabetes, cardiovascular disease, even some cancers. Plus, obesity often involves chronic low-grade inflammation.
Inflammation from fat.
Yeah, that ectopic fat isn't just sitting there. It's metabolically active and releases inflammatory signaling molecules. This attracts immune cells, like macrophages, into the tissue, which further fuels inflammation and metabolic dysfunction. It really becomes a vicious cycle. Inflexibility leads to fat deposition, which causes inflammation, which worsens inflexibility.
Wow. And inflammation itself is tied to metabolism.
Deeply. The sources mention this fascinating idea of trained immunity. It turns out that innate immune cells, like macrophages, which we used to think had no memory, can actually be trained by certain triggers to respond more strongly later on.
Trained immunity, how does that work?
Well, it has a metabolic basis. For example, if you stimulate certain immune cells with bacterial components, they undergo a dramatic metabolic switch. They shift from efficient energy production, oxidative phosphorylation, to rapid glycolysis, burning glucose really fast, kind of like cancer cells do.
Why would they do that?
It seems necessary for them to mount a really robust inflammatory response. They need quick energy and building blocks. And this switch involves many of the same metabolic pathways we've been talking about: ACT, mTOR, HIF-1 alpha, even sirtuins.
It's all connected.
It really is. And in fat tissue specifically, you find different types of macrophages. There are M1 types, which are pro-inflammatory, tend to rely on glucose and contribute to insulin resistance. And then there are M2 types, which are more anti-inflammatory, prefer burning fatty acids, and help maintain metabolic health. Studies show you can actually influence the inflammatory state of these macrophages by tweaking their metabolism. It highlights just how intertwined immune responses and metabolic pathways are.
Okay, what about cardiovascular disease? How does flexibility fit in there?
The heart is another incredibly metabolic organ. It usually loves burning fatty acids. That's its main fuel under normal conditions. But it absolutely needs to be flexible.
Right.
Because during periods of high demand, like strenuous exercise, the healthy heart needs to quickly switch gears and start burning glucose and lactate more effectively to keep up ATP production. This switch involves signals like increased calcium inside the cells and activation of the sympathetic nervous system, the fight or flight response. These signals boost the machinery for glucose uptake and burning.
So flexibility is key for heart function.
Absolutely. When metabolic inflexibility sets in, maybe because excess fat starts accumulating within the heart muscle cells themselves, that's called cardiomyocyte steatosis, it can lead to all sorts of problems: abnormal signaling, oxidative stress, mitochondria not working properly. These can contribute to heart failure. Also, nutrient stress itself can impact how the heart uses glucose via those ACT and AMPK pathways.
And cancer, you mentioned that earlier too,
Yeah, cancer is notorious for hijacking metabolic pathways. You've probably heard of the Warburg effect.
where cancer cells prefer glucose even with oxygen.
Exactly. They often rely heavily on glycolysis, fermenting glucose even when oxygen is available, which is normally inefficient. But it's not just that they're stuck on glucose. Many cancer cells actually display a kind of pathological metabolic flexibility. They become very adept at switching between fuels, glucose, fatty acids, amino acids like glutamine, depending on what's available in their local environment or what nutrients are scarce. This adaptability helps them survive, grow, and resist treatment.
So they use inflexibility to their advantage.
Or rather, a distorted kind of flexibility. And researchers are now actively exploring ways to target these metabolic vulnerabilities, this metabolic adaptability of cancer cells, as a therapeutic strategy. Some aggressive tumors even figure out how to make certain essential nutrients themselves, like the amino acid glutamine, which makes them even more self-sufficient and harder to target.
It's incredible how central metabolism is to all these major diseases.
Mhm.
We've focused a lot on diet and calorie intake, but the sources mention other factors influencing flexibility, right? Things beyond just food.
Oh, yeah, definitely. It's not just about what's on your plate. The research points to several other really interesting influences.
Like what? Give me an example.
Temperature is one. Think about animals adapted to cold environments. Certain birds, fish, animals that hibernate. They show amazing metabolic adaptations.
How so?
Well, birds acclimated to the cold, for example, dramatically boost their ability to burn fat to generate heat. They increase fatty acid transport into cells, ramp up mitochondrial numbers, and fat-burning enzymes. Hibernators are even more extreme. They switch almost entirely to using stored fat to fuel their bodies through months of super low metabolic rate. Key growth pathways like ACT and mTOR basically get shut down.
Does this apply to humans at all?
Well, the sources cautiously mention the idea that maybe, just maybe, reducing core body temperature slightly in mammals like us might have some beneficial metabolic effects, but that's complex and needs way more study. What's more relevant, perhaps, is brown adipose tissue, or BEAT.
Brown fat, the good fat.
The good fat, yeah. It's specialized fat that burns energy to generate heat. It gets activated by cold and also by our sympathetic nervous system. Interestingly, people who are obese tend to have less active BEAT, and low BEAT activity is linked to problems like type 2 diabetes and even aging.
Can we activate our brown fat?
Exposing people to cold environments can activate BA. It can even potentially trigger a process called browning, where some of our regular white fat starts acting more like brown fat. And this seems to improve insulin sensitivity.
So, cold showers.
Maybe, although the effect size and practicality are debated. Trying to activate it with drugs, adrenergic therapies, hasn't been super successful and often comes with cardiovascular side effects. So cold exposure is interesting, but maybe not a magic bullet.
Okay, what else besides temperature?
Your body's internal clock, your circadian rhythm, this connection is huge.
Our sleep-wake cycle.
Exactly, but it's much more than just sleep. Our internal circadian system profoundly influences metabolism. It coordinates the timing of everything when lipids are processed, the daily rhythm of mitochondrial function in different tissues. These internal clocks in our organs interact directly with nutrient sensors, control metabolic enzymes, and regulate key gene regulators, many of which cycle over 24 hours.
So timing matters.
Hugely. Key metabolic regulators like AMPK and ACT are themselves influenced by the daily rhythms of when we eat. When the system gets disrupted, think shift work, erratic sleep schedules, eating late at night, it throws metabolic timing out of whack.
Jet lag, but internally.
Kind of, yeah. The circadian misalignment is strongly linked to metabolic inflexibility, insulin resistance, and a higher risk for obesity, type 2 diabetes, and heart disease. Figuring out exactly how this disruption causes problems is a really active area of research.
And aging, you mentioned that too. Does flexibility just decline naturally?
It generally seems to, yes. Metabolic flexibility tends to decrease as we get older, and this decline is thought to be a pretty significant contributor to many age-related diseases. So the flip side is targeting metabolic flexibility, trying to preserve or improve it, might be a strategy to extend not just lifespan, but health span, the years lived free from chronic disease.
How does aging affect it?
Well, mitochondrial dysfunction is a known hallmark of aging, and it's closely tied to cells becoming senescent, basically old and dysfunctional, and releasing pro-inflammatory signals. Those key nutrient-sensing pathways we keep mentioning, INSLUM-GF, mTOR, AMPK, SIRTs disruptions in these, are also fundamental features of aging, and they directly impact metabolic health and longevity.
So it sounds like being less active as we age really contributes to this decline.
Absolutely. A sedentary lifestyle often increases with age, and it's a major driver of these age-related metabolic problems.
Yeah.
But there's some good news here.
Oh,
Yeah, one study specifically looked at middle-aged postmenopausal women. They put them through endurance training and found it significantly improved their metabolic flexibility, especially their ability to mobilize and burn fat.
so it's not too late to improve it.
That's the encouraging part. It suggests that even later in life, this crucial metabolic adaptability can still be trained and improved. The sources really hammer home that exercise is a cornerstone strategy, not just for flexibility, but for healthy aging in general.
Okay, let's zoom back in a bit. We've touched on mitochondria, adipose tissue, muscle. Can we talk more about their specific roles based on the sources?
Definitely. Let's start with mitochondria again because they keep popping up.
Central command, you said.
Right, and they're incredibly dynamic. They're constantly changing shape. They can split apart, fission, or fuse together, fusion. And this shape-shifting, their morphology, seems to be influenced by how much fuel is available.
Shape matters.
It seems to. These changes in shape appear to affect how efficiently they can carry out oxidative phosphorylation and how they prioritize fuels. Some studies looked at mitochondria isolated from animals fed high sugar or high fat diets, and they found reduced flexibility just within the mitochondria themselves.
So it's not just signals from the rest of the cell.
It suggests it's partly an intrinsic property of the mitochondrial network itself. Of course, it's regulated too, rapidly by things like calcium levels or chemical tags added to proteins, and longer term by turning genes on or off that control how many mitochondria a cell has.
You mentioned nutrient overload changes their shape.
Yeah, nutrient overload tends to promote fragmented networks, lots of smaller mitochondria. Some researchers think this might actually be protective, maybe slowing down the processing of that overwhelming fuel supply. Conversely, calorie restriction often leads to more interconnected, elongated networks, which are generally associated with higher efficiency in making ATP.
So lots of links between mitochondria and inflexibility.
Tons of links. But the sources are careful to note that proving a direct causal link showing definitively that mitochondrial dysfunction causes inflexibility, rather than just being associated with it, is still something researchers are working hard to nail down.
Okay. And mitochondria do more than just energy.
Oh, much more. They're involved in regulating fat synthesis and glucose production in the liver, controlling fat release from adipose tissue, influencing insulin secretion by pancreatic beta cells, even affecting the production of those adipocyte hormones by fat cells. They're critical players at so many levels.
Which makes sense why problems there are linked to diabetes and obesity.
Exactly. And why exercise, which boosts mitochondrial performance, is linked to better flexibility and insulin sensitivity.
What about adipose tissue itself, beyond just storage?
Adipose tissue is hugely important. Healthy, functional fat cells are actually protective. They help soak up excess lipids, reducing the burden on other tissues like the heart and liver, preventing that harmful ectopic fat accumulation. They also maintain a healthy balance of those signaling molecules, the adipokines.
Like adiponectin.
Adiponectin is a key one. Higher levels are generally good news, linked to better adipose tissue function and overall metabolic flexibility. It acts on the liver and muscle to improve fuel handling. Leptin is another major player, signaling to the brain about energy stores. Though in obesity, the brain often becomes resistant to leptin's signal.
So fat tissue is sending and receiving signals.
Constantly. It's both responding to and influencing metabolic, inflammatory, and hormonal signals throughout the body. One study even found that increasing adiponectin helped promote healthy blood vessel growth within fat tissue itself during diet-induced obesity, which is thought to be beneficial.
And muscle. Not just for movement, you said.
Right, muscle acts like an endocrine organ too. When muscle contracts, especially during exercise, it releases these signaling molecules called myokines.
Myokines.
Yeah, they travel through the blood and influence metabolism in other tissues, fat, liver, pancreas, even the brain. They can affect glucose disposal, fat burning, fat breakdown. Myostatin is one example. It normally inhibits muscle growth, but it also affects fat tissue. Exercise lowers myostatin, obesity raises it. Knocking out myostatin in mice actually protects them from diet-induced obesity and improves their insulin sensitivity.
Interesting. Any other important myokines?
Meteorin-like is another one that's released from muscle with exercise and also from fat tissue with cold exposure. It seems involved in energy balance and inflammation, but its exact role and potential are still being worked out. Then there's irisin.
Ah, yes, irisin,
Mm,
the exercise hormone.
that was the initial excitement, yeah. Proposed as a myokine that could improve glucose regulation. But the human studies have been really contradictory. One source even noted a study in pigs where exercise training didn't increase the precursor protein for irisin in their muscles, casting more doubt on its straightforward role, at least in that context. So the jury's still out on irisin in humans.
Okay. The sources also mentioned specific studies on hormones.
They did. One used human muscle cells in a dish and found that high, maybe even super physiological doses of estrogen or testosterone actually decreased glucose metabolism specifically, the conversion of glucose to stored glycogen.
Decreased it. That seems counterintuitive.
It does. The effects seemed linked to activating a signaling pathway involving PKC. It suggests these hormones can influence these pathways, but we definitely need more research using normal physiological levels to understand the real-world implications.
What about estrogen and obesity together?
There was a rat study looking at that. Removing the ovaries, which removes estrogen, impaired insulin sensitivity, especially in the brain. Giving estrogen back helped improve insulin sensitivity, but only in the rats eating a normal diet. It didn't help the obese rats that had their ovaries removed.
So obesity negated the estrogen benefit.
It suggests that the combination of obesity and estrogen deficiency creates a more severe or perhaps different type of insulin resistance that estrogen replacement alone can't fix. It really highlights how multiple factors interact.
And leptin. Any updates there?
A mouse study showed that treating obese mice with leptin actually improved mitochondrial function. It helped restore struggling liver mitochondria and boosted muscle mitochondria. It also reduced liver fat. So, in mice at least, leptin seemed to have positive effects on mitochondria and fatty liver in the context of obesity. Needs human confirmation, of course.
Did leptin affect fish when You mentioned that earlier.
Ah, right. Another study looks specifically at that. Fetuin A is linked to insulin resistance. But this research, using both human data and cell studies, found no evidence that leptin influences Fetuin A levels. Neither calorie deprivation nor leptin therapy changed Fetuin A, and leptin didn't affect its gene expression in cells. So it seems those two pathways are separate.
Okay. And leptin and the immune system.
One more interesting finding, an in vitro study showed leptin directly regulated autophagy, that cellular recycling process in human T cells, which are key immune cells. Leptin actually inhibited autophagy, acting via the mTOR pathway. Mice lacking leptin had higher T cell autophagy. This provides a direct link between leptin, nutrition levels, and immune cell function.
Fascinating. Okay, this is all incredibly detailed science. How do researchers and clinicians actually try to measure metabolic flexibility in people? And are there any new biomarkers emerging?
Yeah, measuring it accurately, especially in a clinical setting, is definitely a challenge. There are some traditional markers and then newer approaches trying to capture this adaptability.
Like basic blood tests.
Some standard markers are still useful indicators of overall metabolic health. One study looked at common liver enzymes, ALT and GGT, in young adults. They found levels correlated with current cardiometabolic risk factors, heart rate, blood pressure, cholesterol. But more than that, those baseline liver enzyme levels actually predicted future changes in weight and body size over 2 years.
So those simple tests are quite informative.
It can be, yeah. They suggest monitoring liver health is important for overall metabolic health surveillance. In that same study, Fetuin A correlated with insulin resistance and blood pressure at the time, but unlike the liver enzymes, it didn't predict those future changes in body size, so maybe less predictive, at least in that cohort.
What about irisin as a biomarker? Still uncertain.
Still quite uncertain for glucose regulation in humans. One study did find a correlation between irisin and higher energy expenditure in some people. Perhaps more promisingly, another study found lower irisin levels in patients with fatty liver disease compared to healthy controls. It was also linked to liver inflammation. So maybe irisin levels could relate to liver health, but it's not clear-cut. The idea of irisin resistance also complicates things, and maybe the body makes it but doesn't respond.
Any other potential biomarkers?
Almontin-1 is another cytokine being looked at. One study found an interesting inverse relationship in people with higher cardiovascular risk. As irisin levels went down, almontin-1 levels went up. Almontin-1 was also linked to less healthy cholesterol particle size.
Okay.
So both irisin and almontin-1 are being explored as potential cardiovascular risk markers, and maybe they interact somehow.
Beyond blood tests, what about imaging?
Imaging is getting better, especially for things like fatty liver. Liver biopsies are invasive.
Right.
So sources highlighted non-invasive techniques like multi-echo MRI and MR spectroscopy, MRS. They tested these in rat livers and found they were actually more sensitive and accurate than looking at tissue samples under a microscope for detecting liver fat induced by high sugar fat diets. And the amount of fat seen on the scans correlated well with insulin levels.
So potential for early detection in humans.
That's the hope. They suggest these non-invasive MR methods could be really valuable, but they need proper validation in human clinical trials.
And for brown fat, how is that measured?
Usually with PTCT scans using a radioactive glucose tracer, 18-FDG. Active brown fat takes up glucose to generate heat, so it lights up on the scan. One study used this to see if low blood sugar hypoglycemia affected B8 glucose uptake during mild cold exposure in healthy adults.
And did it?
Interestingly, no. They found no significant difference compared to when glucose levels were normal. It suggests the relationship between blood sugar levels, beat activity, and overall energy expenditure is probably more complex than we thought. Still more to figure out there.
Okay, this brings us to the really practical part. For people listening, what can they actually do? Based on all this research, what are the most promising ways to improve metabolic flexibility?
Right, the actionables. And the good news is, lifestyle interventions come up again and again as the most powerful tools we have, especially for tackling metabolic syndrome.
Starting with weight loss.
Weight loss is often foundational, yes. The sources consistently emphasize that even modest weight loss, just 5% of body weight, can lead to significant improvements in obesity-related metabolic issues. Losing visceral fat, that fat around the organs, seems particularly beneficial for improving fasting glucose, cholesterol, triglycerides, blood pressure.
And how to achieve that? Calorie restriction.
Energy-restricting diets are the main approach discussed. Creating that calorie deficit forces the body to tap into stored fat. These diets are proven effective in animal models and show clear promise in humans. Intermittent fasting, specifically, showed benefits in rodent studies, improved insulin and leptin sensitivity, more ketones, less fat mass, less inflammation. And caloric restriction, done properly, is still considered the most robust way to extend health span and reduce risks of major chronic diseases in animal models.
But sticking to diets is hard.
That's the huge challenge, isn't it? Compliance is tough. One source made an interesting point. Maybe the constant eat less, move more mantra isn't the most effective framing. They suggested focusing on breaking up sedentary time might be key. Interrupting long periods of sitting with even light activity, or maybe doing exercise snacking, short bursts of vigorous activity, perhaps before meals could potentially be more sustainable and effective for some people than just focusing on formal exercise or continuous dieting.
Exercise snacking, I like that. What about more traditional exercise?
Exercise training is presented as maybe the principal preventative strategy for improving metabolic flexibility and promoting healthy aging. Skeletal muscle is key, obviously, but exercise benefits the many tissues. Regular activity can help shrink fat cells, make fat tissue handle glucose better, and improve insulin sensitivity overall.
Does it affect fat browning?
Chronic endurance exercise in rats did promote that beneficial browning of white fat, yes, although interestingly, that effect was less pronounced if the animals were also eating a high-fat diet. In humans, exercise is known to reduce fat in the liver, and mouse studies suggest a molecule called PGC1 alpha is crucial for that. Exercise also clearly benefits the heart, reducing cardiometabolic risk by improving insulin sensitivity, lowering fat content, and boosting mitochondrial function.
Are there specific programs that work?
One pilot study mentioned the KLAKS program, an exercise-based lifestyle program for obese adolescents. After a year, they saw significant improvements in a range of metabolic risk factors: body fat, BMI, blood sugar control markers, blood pressure, HDL cholesterol, liver enzymes. It suggests comprehensive, sustained programs can definitely move the needle on these comorbidities.
But just breaking up sitting time might not be enough.
One study in kids and teens didn't find benefits for insulin or glucose just from breaking up sitting with light walking. So while reducing sedentary time is likely good overall, we might need more specific strategies to combat its metabolic consequences. Another study looked at a single bout of exercise after a high-fat meal in healthy men.
Did it help?
It didn't completely fix the immediate negative impact of the meal on glucose and insulin, but it did normalize levels of GLP-1, an important gut hormone involved in glucose control, which the high-fat meal had messed up. So a single session can mitigate some effects, but probably consistent exercise is needed for bigger post-meal benefits, especially with high-fat foods.
Okay, what about diet beyond just calories? Does timing or composition matter?
Absolutely. Meal timing can significantly impact flexibility, mainly through those circadian rhythms we discussed. Eating late or erratically, outside your body's natural active phase, disrupts the synchronization. Rodent studies on time-restricted feeding confining eating to a specific window each day showed it protected against obesity and insulin resistance and helped restore the healthy cycling of metabolic genes.
So when you eat, matters. What about what you eat? High protein.
One study looked at high-protein diets, like 2x or 3x the RDA, during calorie restriction. Surprisingly, it did not prevent the loss of lean muscle mass any better than normal protein, nor did it change key hormone levels involved. So, at least in that study, extra protein didn't seem to be the magic bullet for preserving muscle during weight loss.
What about fats? Balanced high fat.
An interesting study gave obese premenopausal women a balanced high-fat diet equal parts saturated, monounsaturated, polyunsaturated for 16 weeks. They actually saw reduced body fat, increased lean mass, and lower inflammatory markers. Specific fatty acids seem to have slightly different effects on inflammation. It suggests type of fat matters, and maybe a balanced high-fat approach, it could have benefits, at least in that group. Needs more study.
Any specific foods? Walnuts, fish?
A walnut-enriched diet, about 43 D-Day, in older adults lowered bad cholesterol, non-HDL and ApoB, which is good for CVD risk, but it didn't impact insulin resistance or inflammation markers in that study. A fish-based diet, high in omega-3s, improved blood vessel function in postmenopausal women with type 2 diabetes. Interestingly, the benefit didn't directly correlate with measured omega-3 levels, hinting maybe other components in the fish were also important.
Mediterranean diet.
Always comes up. In men with metabolic syndrome, just eating a Mediterranean diet without losing weight didn't change most adipokine levels. But after they lost weight through calorie restriction, then the Mediterranean diet led to better changes in leptin and adiponectin compared to a control diet. It really underscores that weight loss itself is often a critical factor for seeing those hormonal benefits from diet changes.
Flaxseed oil versus olive oil.
A study in healthy young adults found both lowered total and LDL cholesterol, similarly from baseline. No major differences between them for adiponectin, inflammation markers, or other lipids. So both seem beneficial for lipids and inflammation, but maybe not dramatically different from each other in that context.
You mentioned even the type of calorie restriction matters. Low fat versus low carb.
Yes, at the muscle level it seemed to. Obese individuals lost similar weight and had similar overall insulin sensitivity improvements on 30% calorie-restricted diets, whether low-fat carb or high-fat low carb. However, the low-fat carb diet specifically improved insulin signaling pathways within the skeletal muscle cells more effectively. The high-fat low carb diet, in contrast, led to some changes in muscle that could potentially promote insulin resistance there. So the macronutrient balance might impact different tissues differently.
Diet versus exercise. Which wins?
A mouse study offered an interesting perspective. A high-fat diet increased fat in the muscles. Switching the mice to a low-fat diet reversed this buildup. But surprisingly, adding exercise did not enhance this reversal in the mice. It suggested that for reducing muscle fat specifically in that model, diet change was the dominant factor over exercise. Needs checking in humans, of course.
One more dietary angle. Methionine restriction.
Yeah, restricting the amino acid methionine in obese mice showed impressive results. Reduced body weight, liver fat, insulin resistance, and inflammation markers. It also seemed to boost the export of fat from the liver, so it suggests targeting specific amino acids could be a strategy, but again, needs careful human trials.
Okay, beyond lifestyle, what about pharmaceuticals? Are there drugs that target flexibility?
Yes, many existing and experimental drugs target key metabolic players, and many of them impact mitochondria or the pathways regulating fuel choice.
With the PPRs?
PPRs are a major target family. PPAR alpha agonists, like fibrates, are used for high lipids and insulin resistance. They boost fat-burning enzymes. PPAR beta-delta agonists look promising for improving fat metabolism, insulin sensitivity, and mitochondrial activity. Tesaglitazar hits both alpha and gamma. Then there are the thiazolidinediones, or TZDs, like rosiglitazone, which activate PPAR gamma. They lower blood sugar effectively by improving glucose uptake and storage, especially helping fat tissue buffer lipids away from other organs.
But they have side effects.
Yes, the TZDs, particularly rosiglitazone, were linked to increased heart attack risk, which really highlighted the potential downsides and complexity of targeting these broad regulators.
What about MTOR?
Manipulating MTOR is also tricky. It's involved in so much, with lots of feedback loops. Completely blocking it with high doses of drugs like rapamycin can cause significant side effects and impair tissue maintenance. Research is ongoing to find ways to modulate it more subtly or develop rapalogs with better safety profiles.
And diabetes drugs like DPP-4 inhibitors.
Right, like sitagliptin. They work by boosting GLP-1 levels. A study showed sitagliptin not only lowered glucose, but also significantly reduced inflammatory markers like CRP and IL-6 in adults with type 2 diabetes. The increase in GLP-1 correlated with the decrease in inflammation. This suggests these drugs might have anti-inflammatory benefits beyond glucose control, potentially useful for other metabolic diseases, though more study is needed there too.
There's a lot happening on the pharmaceutical front as well.
Definitely. It's a very active area, trying to find ways to pharmacologically restore some aspects of metabolic flexibility or mitigate the consequences of inflexibility.
This field seems incredibly dynamic, with so much still to uncover.
It really is. And researchers face some significant methodological challenges.
How so?
Well, we rely heavily on model organisms, yeast, worms, flies, rodents, primates, to understand the basic pathways. That's crucial. But translating findings to humans isn't always straightforward.
Rodents aren't tiny humans.
Exactly. They differ metabolically in key ways. Plus, results can vary depending on the specific strain of mouse, whether they're male or female, the exact experimental setup, the techniques used. There isn't always a perfect gold standard method for assessing flexibility in humans either. Even things like cage density or room temperature can affect rodent metabolism.
And it's just so complex, right?
That's the biggest challenge. So many interacting mechanisms influence metabolism. Isolating the causal factors driving inflexibility, proving X causes Y, rather than just being associated with it, is really tough.
So how do researchers tackle that complexity?
Increasingly with what's called a crossomics approach, trying to integrate huge data sets, genomics, transcriptomics, proteomics, metabolomics, phenomics, to get a much more holistic picture of what's changing across the entire system.
Using big data.
Essentially, yes. And coupling that with systems biology and computational modeling, scientists build mathematical models of metabolic networks.
Like a virtual metabolism.
Kind of. These models help identify critical points, predict how the system might behave if you change something, and even pinpoint potential drug targets or biomarkers. For instance, one model suggested insulin resistance in one tissue inevitably causes knock-on problems elsewhere. Another model of muscle metabolism supported the idea that competition between fats and glucose at that key PDH enzyme is important in insulin resistance. These tools are becoming vital for making sense of the complexity.
So, looking ahead, what are the big hurdles and hopes for translating all this knowledge?
The big hurdle is bridging that gap between the vast amount of preclinical data showing flexibility is important and developing truly effective, applicable interventions for humans. Because flexibility operates at so many levels, substrate, cell, tissue, organ, whole body, and is affected by so many factors, it's a really challenging target.
What's needed to move forward?
The sources really call for a coherent, multidisciplinary approach, bringing together all those omics experts, plus enzymologists, molecular biologists, bioinformaticians, nutritionists, clinicians, and crucially, putting a much stronger emphasis on well-designed in vivo human studies. We need to move beyond just associations found in lab models.
More human trials.
Exactly. And better tools, faster, more accurate metabolomics, combining different methods, developing platforms specifically focused on translating findings to human relevance. The ultimate hope is that with better understanding and tools, we can move towards more personalized analysis and tailor interventions to an individual's specific metabolic profile.
It really does sound like, even with everything we've unpacked today, we're still just scratching the surface in many ways.
Absolutely. It's a fundamental aspect of our biology, so critical for just daily living and clinical care. The field is rapidly growing as we appreciate just how central metabolic health is.
Wow. Okay, that was genuinely a deep dive into metabolic flexibility. We started with that seemingly simple idea of your body switching fuels, went right down into the weeds of the biochemical dance in the mitochondria, saw how losing that adaptability is tied into huge health problems like obesity, diabetes, heart disease.
Yeah, the connections are stark.
And even explored how things you might not immediately connect, like your sleep schedule or the ambient temperature, can play a significant role.
And we covered the really exciting potential of interventions, too, from powerful lifestyle changes, diet composition, meal timing, different types of exercise, breaking sedentary time, and all the nuances there, to the ongoing quest for effective drugs. But the takeaway is clear. It's a complex system from the tiniest organelle right up to the whole organism, and we still need a lot more research to fully understand it.
So for you listening right now, what's the bottom line? Well, it definitely reinforces that maybe simple advice like eat less, move more, while directionally correct is way more complex in
Oh,
practice.
sure.
It also really highlights how seemingly unrelated things, getting good sleep, managing stress, which impact those circadian rhythms and inflammatory pathways we talked about, are probably just as crucial for your metabolic health as what you eat or how much you exercise.
Which leads to a final thought, maybe something for you to consider. Given how absolutely vital metabolic flexibility seems to be for healthy aging, for preventing so many common diseases, and acknowledging that the science is still working hard to establish all the causal links and translate findings from labs and animals into foolproof strategies for humans, what's one small but evidence-backed step you could realistically take, starting today, based on some of the things we discussed, to support your body's own innate ability to adapt and manage energy effectively? Just something to reflect on as you navigate your own energy balance and health journey.
That's a great question to end on. Thanks for joining us for this deep dive. We'll catch you next time with another stack of sources to unpack.